Welcome to the PubMed Daily Review for March 05. Today we scanned 58 new open access papers. Here are the top 5 selected for detailed analysis. Up next, our first paper out of 5, Mitochondrial transfer from glia to neurons protects against peripheral neuropathy by Jing Su and Verong Ji from Translational Pain Medicine, Department of Anesthesiology, Duke University Medical Center, Durham, North Carolina, USA. Let's break down its key contributions. Primary sensory neurons have incredibly long axons, stretching up to a meter. This means they have a huge energy demand, and keeping their mitochondria healthy is crucial. When mitochondria fail, it can lead to peripheral neuropathy, a condition seen in diabetes and after chemotherapy. We know satellite glial cells wrap around these neurons, but how exactly they support them, especially regarding mitochondria, remained a mystery. To investigate this, the researchers used a multi-pronged approach. They started with cell cultures, observing how glial cells transferred mitochondria to neurons. They then moved to ex vivo DRG tissue and finally to in vivo mouse models, using genetic tools to label mitochondria specifically in glial cells. Advanced microscopy techniques like CM and TEM were used to visualize the structures involved, like tunneling nanotubes, and pharmacological agents were used to block specific transfer pathways. The results were striking. In living mice, they observed significant transfer of glial mitochondria to sensory neurons over time, with over 20% of neurons receiving these vital organelles by day 10. Blocking this transfer with drugs like cytocalicin B significantly reduced the process. Importantly, in models of diabetes, this protective transfer was impaired, and conversely, after nerve injury, neuronal activity increased the demand for in-transfer of these mitochondria. This study reveals a critical, previously unrecognized pathway for maintaining neuronal health. It highlights how satellite glial cells directly support sensory neurons by donating mitochondria, and suggests that problems with this transfer are involved in conditions like diabetic neuropathy. This opens up exciting new avenues for developing therapies aimed at boosting this natural protective mechanism to treat neuropathic pain. Up next, our second paper out of 5. A general genome editing strategy using CRISPR lipid nanoparticle spherical nucleic acids by Zhenyu Han and Chad A. Merkin from A Department of Chemistry and International Institute for Nanotechnology, Northwestern University, Evanston, Illinois 60208. Let's break down its key contributions. CRISPR gene editing holds incredible promise for treating genetic diseases and cancer. However, getting the CRISPR machinery into cells effectively has been a significant challenge. We need ways to deliver these complex genetic tools without causing harm or triggering unwanted immune responses. This study explores a new approach to overcome these delivery barriers. To tackle the delivery problem, the researchers developed a novel system called CRISPR lipid nanoparticle spherical nucleic acids, or LNPSNAs. They compared these LNPSNAs to traditional lipid nanoparticles, or LNPs. The key difference is the LNPSNAs have a special DNA shell on their surface. They tested how well these particles got into cells, how toxic they were, and how effectively they could edit genes, including precise edits using donor DNA. The results were quite impressive. The LNPSNAs showed a 2 to 3-fold increase in cellular uptake compared to regular LNPs. This led to a similar 2 to 3-fold boost in gene editing efficiency, meaning more indels were created. Crucially, when trying to make precise edits using donor DNA, LNPSNAs achieved a 21% repair rate, more than double the 8% seen with standard LNPs. They also found these new particles were less toxic. This study introduces a powerful new tool for gene therapy. The CRISPR LNPSNAs represent a significant advancement in delivery technology, showing better efficiency and safety. This could pave the way for more effective treatments for a range of genetic conditions. While these findings are exciting, future research will focus on testing these particles in living organisms and ensuring their long-term safety and manufacturability. Up next, our third paper out of 5, Prevalence and Outcomes of Frailty in Older Men. The Manitoba follow-up study by Philip D. St. John and Robert B. Tate from Section of Geriatric Medicine, Max Rady College of Medicine, University of Manitoba, Winnipeg. Let's break down its key contributions. Frailty is a growing concern in our aging population, characterized by decreased resilience and increased vulnerability. While we know it's linked to worse health outcomes, we need a clearer picture of how common it is and what factors contribute to it, especially in older men. This study followed over 1,700 older men from the Manitoba follow-up study. Researchers constructed a frailty index using medical, functional, and psychosocial data collected over many years. They then tracked these men to see who died or required long-term care. The study found that frailty, dementia, and the need for long-term care all increased significantly with age. Importantly, a higher frailty index was consistently associated with a markedly higher risk of mortality and long-term care admission, even at advanced ages. While functional impairment was a strong predictor, the impact of medical conditions lessened in very old age. This research highlights that frailty is not just a marker of aging, but a significant risk factor for poor outcomes in older men. Clinicians should assess frailty and adapt care accordingly, shifting focus towards functional support in very old age. Further studies are needed to understand the dynamic nature of frailty over time. Up next, our fourth paper out of 5, Intratumoral Preservation of the Stem-like Malignant Cell Proportion in Glioblastoma, Prognostic Impact, and its Radiomethylomic Signatures by Dashiell J., Durtu and Tariq K. Youssef from University of Pennsylvania. Let's break down its key contributions. Glioblastoma, the most aggressive brain tumor, is notoriously difficult to treat due to its significant internal diversity. While we know this heterogeneity exists, how the proportion of stem-like cancer cells varies within a single tumor and its impact on patient outcomes has remained unclear. This study aimed to shed light on this by investigating the consistency of these stem-like cells and exploring if we can predict their presence using advanced imaging techniques. To tackle this, researchers analyzed DNA methylation profiles from a large set of glioblastoma samples, including multiple samples from the same tumor to assess consistency. They employed sophisticated deconvolution techniques to estimate the proportion of stem-like versus differentiated malignant cells. Furthermore, they integrated these findings with detailed information extracted from multiparametric MRI scans, creating what they call radiomethylomic models to see if imaging could predict these cellular states. The study revealed a striking finding. The proportion of stem-like malignant cells was remarkably consistent across different samples from the same tumor. Importantly, tumors with a higher proportion of these stem-like cells were associated with significantly shorter overall survival. This also meant patients with higher stem-like cell proportions experienced less benefit from a common treatment related to MGMT promoter methylation. The radiomethylomic models, combining MRI data with methylation insights, showed promise in predicting this crucial tumor characteristic. So, what does this mean for patients? This research establishes that the proportion of stem-like cells is a consistent and important characteristic of a glioblastoma tumor, directly impacting survival. The development of radiomethylomic models is particularly exciting, as it suggests we might be able to predict these cellular states non-invasively using MRI scans. Ultimately, this work could lead to more tailored and effective treatment approaches for individuals battling glioblastoma. Up next, our fifth paper out of 5, Atomic Force Microscopy-Based Photothermal Infrared Microscopy for Aqueous Environments Using Graphene-Based Microfluidic Cells by Yasuhiko Fujita and Hiromi Watanabe from A. Research Institute for Sustainable Chemistry, National Institute of Advanced Industrial Science and Technology, AAST, Kagomiyama March 11, 32, Higasha Hiroshima, 7390046, Japan. Let's break down its key contributions. Understanding how polymers behave when hydrated is key to many fields, from biology to materials science. However, studying these materials in water using infrared microscopy has been a significant challenge. This paper introduces a new technique that aims to solve this problem. The researchers developed a novel setup using atomic force microscopy combined with photothermal infrared spectroscopy. They employed microfluidic cells with a graphene layer, which acts as an ultra-thin, infrared transparent window. This innovative design allowed them to probe hydrated polymers directly in water. The results were quite promising. The team was able to successfully detect changes in polymer structure, specifically swelling, by analyzing the PTIR spectra. This demonstrated that their novel method works effectively even in aqueous conditions, opening up new possibilities for studying hydrated materials. This research represents a significant advancement in our ability to study hydrated materials at the nanoscale. By enabling chemical imaging in aqueous environments, this technique has broad implications for understanding biological systems and developing new materials. Further applications in areas like drug delivery and biomaterials are highly anticipated.